Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Precision for Medicine webinar titled Illuminating Immunohistochemistry, Right-Sizing Your Validation and Regulatory Strategy. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host. Today, we're very fortunate to be joined by Dr. Kennon Daniels and Ms. Christie Bongel, who will be in conversation throughout this presentation to discuss their collaborative approach to IHC validation and IVD regulatory strategy. Dr. Daniels is VP of IVD Regulatory Consulting at Precision for Medicine, specializing in developing U.S. regulatory strategies and FDA submissions for IVD assays and instruments, and has years of industry experience in all aspects of product development, analytical and clinical trial protocol designs, Q submissions, IDEs, de novos, 510Ks, and pre-market approval platforms. Ms. Christie Bongel is a site director for Precision for Medicine's IHC Center of Excellence. Ms. Bongel brings years of experience as a leader in scientific operations, cytology, laboratory medicine, and molecular diagnostics. She excels as a collaborative partner that focuses on building an energized team dedicated to delivering life-changing outcomes for patients. Today, Dr. Daniels and Ms. Bongel will provide a comprehensive overview of applicable laws and regulations. They will guide you through mapping your U.S. regulatory path and the utilization of assays in clinical trials. Additionally, they will navigate the regulatory differences between the U.S. and EU and discuss key considerations for IHC assay development and validation. And without any further ado, I'm pleased to welcome our first speaker, Christie Bongel. Christie, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you for the introduction, Liam. Before we start, I'd like to provide a quick introduction to Precision for Medicine. Precision for Medicine is, as our name implies, a precision medicine clinical research organization with a breadth of solutions, including biospecimens, specialty and central lab services, and full-service clinical trials. We apply numerous technologies and provide expertise to address complex challenges with the goal of helping life science companies improve the speed, cost, and ultimately success of bringing life-changing therapies to patients. We provide this support globally. As you can see here, we have 18 offices in North America, Europe, and Asia Pacific, and 7 specialty labs across North America and Europe. We're actively running clinical trials in over 50 countries, managing sample logistics in more than 55, and have more than 25 years of experience in running clinical trials. Our IVD regulatory team has vast experience with over 200 submissions for IVD and CDX, Q submissions, IDE applications, and Annex 14 submissions, as well as over 250 510K, EUA, de novo, and PMA submissions for both IVD and CDX in the US. Overall, Precision is well positioned globally to support the development of diagnostics and therapeutics. Now we'll turn over to Kennon to go over a few applicable laws.
Thanks, Christy. So we just wanted to level set the audience as far as language, acronyms, things that we're going to use throughout the presentation to discuss IHC assay development. Just give you a little background so that when we reference this downstream, you have a little understanding of what we're talking about. So we're going to talk a lot about CLIA and the CLIA regulation in the US, as well as the CLSI guidelines, which are Clinical Laboratory Standards Institute guidelines, as well as the IVDR, which is the in vitro diagnostic regulation, which was implemented in Europe in May 26th of 2022. Then we'll also talk a little bit about overall quality systems. So the quality system regulation in the US is 21 CFR Part 820. This quality system talks about good manufacturing practices, and of note, and the FDA has issued a new rule regarding quality system where it's going to now be called the Quality Management System Regulation, which will be implemented in 2026. And this new rule kind of merges the 21 CFR Part 820 with ISO 13485, which is an international standard that organizations used regarding the design, production and development of manufacturing of medical devices. We'll also talk a little bit about ISO 15189, which is a standard for medical laboratories in Europe and a requirement of sample testing in the EU, and ISO 14971, which talks about risk management of medical devices as part of the design control process. So these are just standards that companies can comply with, as well as for Europe and the US.
So Kennon, when a client comes to you saying they're interested in potentially taking an IHC to a CDX, what are the first thoughts and questions that come to your mind?
So normally when we speak with clients and they have an assay that could potentially be used for a companion diagnostic, we like to talk about what is their overall global strategy for the assay, their roadmap, how are they using the assay as part of the clinical studies? Is it for research? Is it for inclusion, exclusion? Is it monitoring the use of the therapeutic? All those questions and answers will inform the regulatory strategy for when you communicate with the agency to develop the appropriate data packages to support regulatory submissions. This slide provides an overview of an ideal pathway, which doesn't always happen, but it kind of walks you through when an assay is being used for an exploratory purpose in phase 1. Then it becomes more for inclusion and exclusion in phase 2, and then finally for the pivotal study in phase 3. And the different regulatory communications that you can have. This is a US focus, so these are all the different communications that you could have with the FDA on the bottom, such as an SRDQ submission or pre-submission to discuss CLSI validation, your IDE application, etc. So when you think about the purpose of the assay, it directly correlates with the level of validation required for that assay use. So if it's an exploratory endpoint or inclusion in the BLA, but not used for directing patient treatment, you're not necessarily going to have to do a CLIA level validation. We don't always recommend that. It's more of a fit for purpose validation or a GXP validation. And then as you move into more of inclusion exclusion with the assay use, that's when you make the associated validation more robust, such as a CLIA level validation. And in some cases for an IDE, the validation requirements are even greater than a CLIA validation. We sometimes call it CLIA plus, where you would have to do some reagent stability, which is not normally part of a CLIA validation. And then if you have a requirement for an investigational device exemption and the assay is considered as high risk, then you would have to do your documentation and development under design control. And then as we progress through commercialization to support that pre-market application in the US, then you would need to do studies to support that analytical validation module as part of the PMA. And those studies are typically designed to align with the CLSI guidelines. These are very robust guidelines to help to guide manufacturers in the actual study designs required for PMA approval by FDA.
So when we think about how that device is going to be used or how that assay is going to be used, right? Your discussion with the client informs their regulatory strategy, which in turn determines our laboratory strategy, right? How rigorous and how in-depth our validation study needs to be. And, I know one of the important things that you said was how the IVD is going to be used in a study. So, what are the potential pathways that this could take?
And that's a great question. There's always a question about risk. How do I know what is the risk to the assay use in the trial? How do I know when it's significant versus non-significant? We get asked that a lot on the regulatory team. And, this slide kind of gives you an overview of different scenarios of how the assay is being conducted, when, such as retrospective or prospective, but the analysis is not being used. It's just informative, so it's more of a research use on the left side, and moving over to the right side. When we consider at Precision for Medicine potential significant risk use of assays, so prospective stratification, enrollment with only IVD positive or negative patients, that's when you would need to do a study risk determination in order to evaluate if an IDE is required.
So who determines significant risk? Is that us?
Well, it's upon the responsibility of the manufacturer, as well as your regulatory team can help assess risk. The evaluation is really based on the context of the investigational study. Are the results being used to determine when the patient receives treatment? Would a false positive lead to the patient not receiving standard of care? What is the safety profile of the therapeutic? Has the patient being included in the trial exhausted all standard of care options? Is the device been used previously? When is the testing conducted? Is there an invasive biopsy or a fresh biopsy being required? That's kind of how you evaluate risk. And there's a lot of options for evaluating risk after you've evaluated risk on confirmation of that risk. So there's no one requirement in the beginning. So there's options of you could do a study risk determination Q submission to the FDA, and the FDA will determine whether or not the assay use for that protocol is significant risk. The risk could be assessed by the IRB. IRB is a FDA surrogate. How you can include a risk assessment in your pre-IND briefing book for oncology indications. You can assume high risk in early stage and just submit the IDE. One thing to note with the IRB submission, if the IRB does not approve your protocol and flags it as significant risk, then you have to go back and talk to FDA, whether it's through the SRD Q submission or an IDE to FDA. Ultimately, FDA is the final arbiter of significant risk, and these risk determinations are irrespective of the phase of the trial, and the risk assessment is independent of your IND submission and approval.
So this is, how does a client determine which path to take? Like if there are all of these options available to them, is it your guidance that guides their decision?
I mean, we look at, the guidance that we provide is based on our own assessment of risk and based on the client's internal timelines and the potential for obtaining a significant risk determination. The risk will have to be assessed and for assay use in each study, especially when the assay is being used for inclusion and exclusion. So we take a lot of different factors into consideration to guide our clients on the best path forward.
So once they've made a decision about what path they're going to take, what role does the IHC assay that we're developing play in informing your submissions?
That's a great question. So typically how we develop the IHC assay, we, in parallel with the laboratory, support the risk assessment, we would typically do an SRDQ submission. But if you look at the slide here, it provides an overview of how the assay is being developed, a fit for purpose assay qualification, validation for using the assay for exploratory testing, with the assumption that this phase 1 study would take approximately 6 months, and then based on the data from the phase 1, the cutoff would be selected, and then the CLIA validation would be designed around that cutoff for use of the assay, using the assay for inclusion, exclusion in phase 2. And while that CLIA validation is being conducted, we would also do the regulatory work on our side, where we would assess risk, submit the SRDQ submission to FDA. The FDA has up to 90 days to review it. The output of that SRDQ submission is either a non-significant risk or a significant risk determination. So if the results of the FDA's assessment is NSR, then the study can begin as soon as that designation is received. However, if the agency provides a SR determination, then we will develop the IDE using the CLIA validation data to submit that package to FDA, and the FDA has 30 days to review the IDE.
So that's a pretty long pathway. I mean, we're looking at, over a year here. And it starts with just assay development and optimization. But, some of our clients come to us and they haven't explored any assays. And so we have to determine what they have in terms of data and what they've done prior to coming to us. Do they have an assay? Do they have an antibody? Is it commercial? Is it proprietary? In other words, are they developing their own antibody? And then we can determine what they still need. Do they have a preferred platform partner, right? There's 3 major IHC manufacturers. That's a consideration. We have all 3 under roof, but we look to our clients oftentimes to guide those decisions. And have they performed any testing already in-house themselves? Do they have information on prevalence within their chosen indication? Have they run any, TMAs? Kennon, you spoke before about all of those tissues that we have here, and that's one of the things that can help in this stage before we move into assay development and optimization, is to utilize those tissues that we have in-house to create TMAs or to perform prevalence testing within a given indication. And then from there, obviously we move through this path that you've shown out and into the CLIA validation ultimately.
So when we talk about CLIA validation, can you help explain what a CLIA validation would look like for an IHC assay?
Yeah, absolutely. Because it's not prescriptive in every case, right? The CLIA regulation that we're referencing here, the 42 CFR 493, is written to apply to any laboratory test, whether that would be a flow assay or PCR or, in this case, IHC, which means that that regulation is written very broadly and there's no definition of how to satisfy each of those performance studies. So we've based our recommendations to our clients upon additional publications and guidance around analytical validations, specifically of IHC assays, as well as reviewing historical CDRH submissions and gathering feedback in concert with your team from the agency. And so, again, this is a bit of a choose your own adventure book, right? Not necessarily a one size fits all approach, but in general, we evaluate that large bolus of tissues within the chosen indication to determine the reportable range, and then evaluate the reference range in a broad subset of both normal healthy tissues as well as neoplastic tissues. From there, our precision and reproducibility studies are designed to cover intra-assay, inter-assay, and inter-operator considerations across both the histotechnicians and the pathologists. We utilize concordance and method comparison studies to satisfy accuracy requirements because the term accuracy is really more appropriately applied when there's a reference standard used, and that just doesn't exist in the technology of IHC. There aren't any calibrators for our instruments. So additionally, we conduct sample stability studies like you were talking about, Kennon. Those can include reagent stabilities or cut slide stabilities, as well as provide scoring development and cutoff determinations to support both a robust test system development and the necessary information for your regulatory submissions. What else do you think about here, Kennon?
I think one potential option for clients is if they have any question about this and they don't know what, in the event that an IDE is required, is their validation enough? Is that CLIA validation going to be robust enough to support the IDE application approval? Then they should talk to the agency through the pre-IDE Q submission process, where you can provide the agency with your study designs and your proposed data package that you will include in your IDE, and it gives you a chance to get feedback from the agency before you conduct any of the studies to make sure that it will be what the FDA is expecting to see as far as data to support that IDE approval. It's really important to streamline that process. And the good news about the Q submission process is it is a free process. It does not cost money for the FDA to look at your data package proposal.
Really?
That is correct.
Huh. How long does it take? What does that add to their timeline?
The pre-IDE process is the agent, if you request a meeting, you would get your feedback for the Q sub within around 70 days. And then you would have your meeting scheduled within 5 days. And after the meeting, then you would make sure to include your meeting minutes and submit those back to the agency within 15 days. So depending on how long it takes you to put that pre-IDE together, it could be 4 to 5 months.
Okay, that's good to think about. And what about, we've talked about the CLIA validation requirements, but what if that's not enough?
Right. So when you think about moving past the CLIA validation and adding to what would be required for a pre-market submission, there's another set of studies that are required, as I mentioned earlier, and these are in compliance with the CLSI guidelines. So there's a guideline for theoretically every type of study that would be needed to validate your assay. And it's very prescriptive on replicates, lots, designs, stats. Acceptance criteria is a big one. You need to set your acceptance criteria before you conduct the studies. And then also thinking through the analytical validation package, it's going to look different depending on your technology. So an IHC assay is going to have a different validation package in the PMA than an NGS assay would. For example, for analytical specificity of an IHC, you're looking at Western blotting, what the staining looks like in normal and tumor tissues, whereas NGS, you're looking at the endogenous and exogenous interfering substances, and you'd have to do an in silico analysis on your primers and probes to make sure you're not having overlap and picking up unintended targets. The FDA also suggests a pre-submission to align on study designs for your CLSI studies to support your PMA. It's another way to talk to the agency, similar to the pre-IDE process. And another consideration, with your analytical validation strategy is where the assay is going to be run. Is it a kitted solution? Is it a single site? The data package will be slightly smaller for a single site, and that you don't have to do a multi-site reproducibility study. Just thinking through all of those components is really important on your overall global regulatory strategy and roadmap.
Yeah, I absolutely agree there. When we're supporting a sponsor through their commercialization plan like this, we actually design our CLIA validation such that it can be added on to support the CLSI validation. And we perform all the studies under design control. So we're building like one comprehensive package to support their pre-market submission. So what if the sponsor isn't operating their clinical trial solely in the US? We've been talking just about the FDA and all of those submissions, but what if they also want to operate their clinical trial in the EU?
Well, there are a lot of differences, honestly, from different product classifications, different quality systems, although the US is, bringing in the ISO 13485, as I mentioned earlier, I think one of the major differences to consider in this scenario is in the US for clinical trials, as I just mentioned, it's about risk assessment. Whereas in Europe, it's about medical purpose and how the assay is being used for a medical purpose. And then instead of an IDE application, if the assay has medical purpose in Europe, then it would require Annex 14 submissions and local ethics committee submissions to be able to use these assays and as part of the clinical trial as future companion diagnostics. In the US, the CDX can be classified as class 2 or class 3. In Europe now under the IVDR or IVD regulation, they are classified as class C. The review in the US is by the FDA, and in Europe, it's done by a notified body. So when we think about medical purpose, under the IVD regulation, which was implemented in 2022, how does the assay use impact the medical management of the patient? If it is impacting the medical management of the patient, then you would have to do Annex 14 submissions to the national competent authorities in each of the individual EU countries. So it's not just one application across the entire European Union. Each individual country has, while the generic content is applicable in all of those countries, there are nuances and different documentation or delivery methodologies that may be different in each country. So it's really important that you know those requirements before you start your clinical trial testing and running the drug studies in those regions, because your assay information needs to be included in the clinical trial application for the therapeutic.
Is it
Next
true,
slide.
Kennon, that in Turkey you have to deliver it by carrier pigeon?
No, it is not a carrier pigeon. It is a hand delivery of the documentation. In Spain, the documentation must be mailed. It is not through an electronic portal, or some countries there are electronic portals where you can deliver that information. So it is definitely a lot of things to manage and different requirements in Europe versus the US now.
It seems complicated.
It can be complicated. If you have the right regulatory team to support you, we can hold your hand and help facilitate the process.
Thank you for holding my hand. So you've talked a lot about the comparisons between the US and the EU, and we think about those two in terms of our assay validations, right? You're looking at it from a regulatory standpoint and what the differences in those submissions are and how those get delivered to the different regulatory bodies, but we're looking at it from the standards from our governments, right? Those are the requirements that we have to meet in terms of our assay validations. And so in the US, as we march through that assay validation in increasing rigorousness and robustness, right? We start out with preclinical or animal studies, xenograft tissue, then move into that GXP validation. Maybe that's RUO, fit for purpose. All of those can be used interchangeably. And then we, ramp up into the CLIA regulations, which have some broad strokes of what the requirements are, and then into the CLSI guidelines, which are, as you said, much more prescriptive in terms of the studies and the number of replicates and all of that has to be included. And in Europe, they're thinking about the same thing, right? They have the GLP, which is, non-clinical as opposed to preclinical. And then they move into GCLP, and they have those ISO guidelines that are supporting them along the path. And then finally, the state-of-the-art or CLSI-like, which is governed by ISO 1345, which, as you noted, is being merged into our own federal quality system. So I think it's important to note that, sometimes we plan to support a sponsor in both the US and the UK or EU. And luckily, we have a sister lab in the UK that we can align and ensure that our validation meets both CLIA and GCLP standards in order to support your submissions both here in the US to the FDA and into the various countries in the EU for their regulatory bodies.
And then as you think about in the EU, the requirements for commercialization, you've walked through the clinical trials, and now you know your CDX is going to be required and you have to commercialize that product. What is that process? What are the requirements? As I mentioned earlier, these are Class C products. There will be technical dossier that needs to be required and developed, and there will be consultation with the EMA. There will be a notified body audit for your QMS, which is similar to the BIMO audit in the US prior to your PMA approval. There's an estimated timeline for CE marking in Europe of around 12 to 18 months. So it's really important to plan ahead and make sure that you allow yourself enough time after the registrational study to get that approval so that you can commercialize contemporaneously. And just continuing that comparison for the US, FDA likes a modular PMA for a CDX approval. While the content is fairly similar as the clinical data and the analytical performance with Europe, there are some differences in requirements for the EU versus US. The timeline, for a modular PMA is going to be a bit longer because the FDA wants to review each module independently and close it out before you move on to the next module. And so this is just an example of a shell of a modular PMA and the order that you could submit the different content for your assay. Whether, it's a good idea to get manufacturing done early because you can start that during your phase 3 or pivotal study and get one of the modules knocked out before you even finish your clinical validation. So it's really important to plan ahead and have your overall timeline and roadmap prepared so you're aware of those timelines and make sure that you can align them with your pharma partner. And so, Christie, holistically, what are some of the main considerations when meeting with a client and understanding their project? What do you think about initially as being the lab or potential lab for supporting their IHC assay development?
Well, one of the things I think is key is that, oftentimes you and I take these meetings together, which is a big part of it. But I think for us, there's really 3 main considerations that we can go through together. First is this assay description, right? That's the lab's main consideration because that's where our expertise shines, and that's really what we're working on in service to that client. But the first thing that we think about is, what's your client or what's your target? Has the client already vetted potential antibodies? Have they been doing some preclinical work in-house, whether that's, ELISA or IHC? Have they looked at commercial antibodies? Are they planning to develop a proprietary one of their own, right? Those are important considerations, both for the lab in terms of what reagents we need to get in-house, but also for you in terms of recommendations for downstream GMP manufacturing. Also, do they have a platform preference? Again, we spoke to the fact that there's 3 major IHC platform manufacturers, and if you're going down the path to eventual kitted solution, it's likely that one of those 3 players will be your partner to perform that kitted solution, that GMP manufacturing. So can we start right away on the platform preference and save ourselves a bridging study at the end, right? That's always a possibility. We can always bridge to another platform, but it saves us time and energy and saves the sponsor cost if they can pick that in the beginning. And what do you think about from an assay point of view?
Just the overall, strategy of how they will want to use the assay. Do they even want a CDX, or they're just looking at the biomarker levels for informative purposes? That we do come across a lot of companies who actually don't want a CDX. They just want to look at levels. at which point it's exploratory, which is a much lower regulatory burden. But as that evolves and you see if the assay would be required for prescribing the therapeutic product, then we think about, the different milestones for talking to the regulatory agencies and what the requirements are in order to help streamline the CDX commercialization process. But it is good to know if, the assay how the assay is being developed, what their target is, and overall understanding of how they intend to use it as part of the trial. And is it quantitative, qualitative, etc.?
Oh yeah, that's a really good point, right? And that takes us to our next point, the data package. We're both putting together data packages, right? We're getting information from the client on any data or information they've already gathered, as we mentioned, on late phase preclinical trials or that prevalence testing across their indication, because that helps us design the validation that's specific to them, right? Depending on, like you said, their desired use of the assay, that really drives the design elements that need to go into the level of validation and how robust it has to be, and what we need to include for any potential data that they have not yet collected themselves. And what other data do we drive into these regulatory submissions?
I would say that the risk determination is a form of data to know that whether or not they need an IDE. That will really significantly affect the timeline. As we showed earlier, you can delay your clinical testing by 3 months, or in the US, and knowing, the data, when the data will be available from your lab to be included in the Annex 14 submissions and local ethics committee submissions in Europe, that those take a bit longer, and each country has different approval timelines. And so just knowing when that data is going to be available can help the overall planning and how much is required, how much time.
Yeah, that's right. And I think the last consideration for us both is the intended use or the indication for use of that assay, right? What is your phased approach? You showed, bringing it all the way back to the beginning, that first slide that covers like phase 1 being used as a retrospective, and then moving into phase 2 where the assay is being used potentially prospectively, and then finally phase 3, your registrational study. And you're using that phase 1 data to inform the cutoff for the phase 2 inclusion and exclusion criteria. And ideally, the sponsor would use that phase 1 retrospective testing to inform the cutoff determinations to move into phase 2, right?
Absolutely.
Yeah.
And having I think for the precision for medicine location at your lab, it's important to know that you have a whole biorepository for tissue specimen to support the CLIA validation or the CLSI level validation, that you have that on hand ready to go, those tissue samples to be able to support that and evaluating the cutoff that would be required based on the phase 1 data.
Yeah, we have done that before. We've had, if they don't find the number of patients that they had intended, we can certainly query our biorepository and backfill with other samples to perform those retrospective testing across the board, across the indication, to kind of try and inform that cutoff decision for them to move forward so that they don't have that kind of delay in moving into their next phase. And we want to think about also the enrollment strategy. What do we think about there in terms of helping inform the client or be responsive
It's
to what their
important.
enrollment strategy is?
Yeah, I think it's really important to understand, how many indications for each patient populations we're evaluating. Is it multiple, or some purely research, or some, used for the inclusion, exclusion, or are you looking at monitoring the response of patients? with the assay. And so it's just understanding how the assay is being used in the clinical trial and that strategy across different indications. It may be different for different indications, and just knowing that what the cutoff feeds the enrollment strategy, because the cutoff determines who is included and excluded. And then that also links into the risk profile, and whether or not the assay use is being considered significant risk or not significant risk, and whether or not an IDE application approval is required prior to testing.
That's right. And part of the enrollment strategy too is going to be geographical, right? As we discussed. Are you going to be solely in the US, or are you going to be elsewhere in the UK or the EU as well? So I think that knowing that information in advance both helps us guide our laboratory strategy in, where we conduct the validations and where we'll conduct the clinical trial sample testing. And it helps obviously inform your regulatory submissions. So Ken, I know you and I have asked each other a lot of questions throughout this. I think it's, informative for both of us and hopefully for everyone in the audience that's been listening to us. But I want to thank everyone for your attention, and hopefully you found this informative. And instead of Kennon and I asking each other questions, we'll move into a Q&A session where you can ask us questions.
Fantastic. Well, thanks so much, Christie and Kennon, for the great presentations today. And with that, we will move on to the Q&A. And as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A tab on the right-hand side of your screen. All right, so we'll start off with a great question here. So do you see the quality of the IHC antibodies, performance and stability increasing, or does that continue to be something that's hard to manage for validating an IHC assay?
Yeah, that's a good question. I think that really we see a marked difference generally in whether the IHC antibody is coming to us from a commercial antibody manufacturer or whether it's a client's own proprietary antibody. We sometimes see quite a bit of variation in terms of concentration specifically when we're dealing with proprietary antibodies, in that they are oftentimes not manufactured to the exact same concentration standard. And so when we are then trying to do lot-to-lot comparisons or things like that, we do sometimes run into challenges. I think that our commercial partners for antibody manufacturing, obviously, especially within the three big players like Ventana and Dako, we see very stable, very consistent performance even across lots. So I think that's maybe the marked difference there in the performance.
Excellent. Yeah, great points. All right, next one here. So with little to no calibration tools for autostainers, do you see the cell line FFPE as a good control material to support tissue controls?
Yeah, that's a great question. I came from a different med tech background prior to coming to histology, and so it was very difficult for me that there weren't calibrators for an IHC stainer. And so we do like cell line controls for that purpose, because you do have kind of a known reference standard, right? And that's what we see IVD manufacturers using as well for their kits. Right, when you order a PDL1 kit, you're getting that control slide that has 3 cell lines on it that are known standards. When you order the HER2 kit, you get that cell line control, right? Because even our manufacturers recognize that that's the most calibrator-like that we can provide within the IHC methodology. The other calibrator that I think we need to find is one for pathologists. So if anybody has one of those, I'd be interested, because I'm sure any of us that are in histology know how hard it is to get your pathologist to agree on something.
Excellent. Another good question here, sort of in a similar vein. Could you explain again how you deal with accuracy and concordance when there isn't a reference standard?
Yeah, that's a good one. I think what we've dug into a lot is CAP recommendations, right? We are a CAP CLIA accredited laboratory. And so even though CAP is focused mainly on clinical laboratories, what we're doing impacts patients no less than a clinical laboratory would. So we've dug into a lot of their supplementary guidelines for IHC analytical validation, and bringing it back to the cell lines. That is one of the recommendations that they encourage for concordance testing, right? They state that accuracy doesn't necessarily isn't well defined within the IHC methodology, and so they recommend concordance, and they offer some suggestions on how to meet that concordance. You can use cell lines as your known standard. You can use a non-IHC based test like Western blot or ELISA or something like that. And we see that a lot of times from laboratories in their submissions to the CDRH, right, Kennon? They're using Western blot information to show that the concordance between the two methodologies. You can also use we use concordance between our labs. Like I mentioned, we have a sister lab in the UK. We operate to the same quality system, the same hardware, the same SOPs. And so we will oftentimes, during the course of a validation, if it's being validated at our lab, we will cut tissue slides that we're using. We'll send them the same lot of reagents, and they'll run it on their instrumentation so that we can confirm, you know, a small multi-site repro that we're getting concordance between two sites. And we find that that also meets the spirit of what CAP is asking us to do and what the FDA is asking us to provide in terms of concordance testing.
Excellent, great answer. All right, another good question here. So how do we ensure our IHC validation meets the various differences in clinical standards and practices across regions?
Oh, I think that's where Kennon and I are hand in hand, right? We rely on her feedback and her team's feedback from their submissions and the intel that they have across all of the various countries. We have a really good understanding of FDA, I think, Kennon, don't you agree?
I think, constantly, getting intelligence from different communications that you have with FDA or other regions based on requirements for inclusion in different regulatory submissions. Ensuring that it meets validation is a strong word. It's based on experience and what we feel like will be a robust validation for the different submissions. I think that's how we stay up to date on what's required and how we know that what we're putting into the package is robust enough.
Excellent, thanks a lot. All right, a few questions related to regulations. So do you have any tips for staying updated with the evolving regulations and guidelines?
Lots of going to FDA roundtables, looking on the FDA website for different published guidelines, overall trainings, webinars, things like that are very helpful. It's an active process that we are continually learning and trying to stay up to date with the fast-paced evolution of requirements and regulations, both in the US and Europe. With the IVDR regulation, we're learning, and it's a process. And as we continue to have these experiences, we're gaining more and more knowledge. But I would just say, be proactive and try to find trainings in different parts of, whether the regions or specifically what you're looking to understand. You can always come to different trainings or going to different conferences where they're talking about what you're looking for. So attending RAPS conferences is always a good idea. They have them in Europe and the US. So It's just a suggestion.
Yeah, some nice ideas. Thanks a lot. Good question here. So why doesn't the EU IVD submission process align with the clinical regulatory submission process? And when do you think this might be fixed?
Yeah, I think, in Europe, I think with UDAMED being implemented, that will help resolve some of the issue to align the clinical and assay specific submissions. Unfortunately, that won't happen for several years, but I think when that is put in place, I think that will help resolve a lot of the questions and hopefully create a more uniform process that's a one-stop shop for submission of documentation.
Fantastic. Good question here. So where do you see digital-based CQS, or computational quantitative scoring, rather than pathologist review, evolving in the next years, and in terms of CDX development and approval?
Yeah, that's one that we discuss a lot with our clients, right? And that Kennon and I are in discussions with on a regular basis, because it's kind of a catch-22 sometimes. We need to use tools and resources that are approved when possible by the FDA for our clinical trial work, right? I'm constantly going to Kennon and going, what's got a 510K clearance within digital scanners or software? So we're constantly looking at those resources that have clearance already, because those are a stronger tool system to be used within a clinical trial. That being said, our clients want to use the most up-to-date things and technologies as part of their systems, right? Whether that's AI algorithms and helping to build those. We have multiple pathologists under roof, so that's oftentimes a service that we provide to our clients is multiple blinded pathologist review of IHC slides in order to help build those ground truth AI algorithms. But those are currently, what we would classify as RUO. Those aren't replacing pathologist eyeballs in a phase 3 clinical trial yet that I'm aware of. And I think Kennon would be in agreement with that. So I think that there is a little bit of a lag in timeline between what we see in the industry and what clients want to use in terms of digital technology and what the FDA has approved and is willing to see in regulatory submissions, right? Kennon, you can speak to this. I think that when we're utilizing something that doesn't already have that 510K clearance, it just puts so much more weight on us to prove that that whole system is appropriate for the use that we're looking at, right? Am I saying that right?
Sort of. I mean, yeah, it does put the burden on our lab to ensure that we're validating the entire system under CLIA. We're bringing it under our own quality system, ensuring that that assay from start to finish is accurate, reproducible, and we validated it appropriately for the use as part of the clinical trial. As far as digital-based scoring, and that is the wave of the future, and I think hopefully that will come into fruition. Right now, as far as clinical medicine, the agency wants to see pathologist interpretation, confirmation of that AI result. And so I think it's in the future. I just, it's especially with companion diagnostics, that's such a big deal on the risk is so much higher when you're prescribing a therapeutic based on a digital interpretation of a slide. So the burden of proof on that manufacturer for who's creating the AI for reviewing that software is very high, and it will be a high data package that will be submitted as part of that PMA submission. So it's just a matter of, working with the agency, making sure it's robust enough and it's definitely reproducible and accurate for the intended use. It's not going to be, you know, across all tumor types initially, right? You're going to have to validate it per indication specifically and robustly.
Well, and I think another thing to think about is all the different factors that have to be included in that CDX package, right? When you think about the existing CDX right now, you can get an IHC kit for PDL122C3, right? And it's got everything you need in it, your reagents, your cell line controls, like we discussed. But when you think about using digital pathology as a CDX, right, you're going to have so many components that have to be in place, from the digital scanner that you use. Only a one has 510K clearance, Kennon? A small handful, right? Oh, yes, the GT450 did just get DX clearance. Thank you. So you only have a couple of scanners, and then the software, and then the test methodology, and then the scoring algorithm, right? So you have so many pieces that would have to all come together for a single CDX that I think it's a pretty big burden of proof right now. And so there may be some delay as the FDA catches up with what we want to do in the industry, right?
Excellent.
That's a great question.
Great answers both. Yeah, thanks a lot. Great discussion. All right, one here about regulatory hurdles. Can you provide any other tips on addressing some potential regulatory hurdles?
I think, there's always
Kennon just calls the FDA.
No, that is not true. So I think addressing regulatory hurdles, the best way is to plan ahead. Most regulatory people are planners. We like timelines. We like to make sure that we have, our initial plan, backup plans, the hurdle of having the time to allow the assay validation, to do the submissions, to allow the back and forth, the questions from different regulatory agencies in order to get your clinical trial enabling submissions, ensuring robust validation packages in your IDE versus your registration, your technical files, or your PMA submissions. So for me, it's allowing that time and planning ahead, and always communicate with the regulatory bodies if you have questions. Don't be afraid to do a pre-submission or a pre-IDE pre-submission. Just allow that time in your development roadmap for your assay or CDX. Don't try to rush it.
Yeah, that's the most important point. Plan for more time than you think you need.
Correct. There's always a delay.
Excellent. I think in the interest of time, we'll make the next question the last one. Are there any guidelines for statistical values to be reported along with IHC analytical sensitivity, specificity, prevalence, reproducibility, or cutoff values?
Yeah, so CAP does provide some of those guidelines. For statistical values, they like to see at least a 90% concordance rate across your studies that you conduct for concordance between the new assay and your comparator assay, or the methodology that you're looking to bring up. And then there's also we create guidelines
to the CLIA validation.
Yes.
Yeah, so in the lens of a CLIA validation, you can refer to those guidelines to support your validation. I think for FDA submission package, I don't know that there's specific statistical values. There's references of how things should be reported in the CLSI guidelines, but it is not specific necessarily to IHC. And so that's why it's really important to understand what performance metrics you want to report, include those in your pre-submission. You can also look at other approvals and see how other companies have reported out their data as a reference to then know what statistics were completed for their approvals. And you can use that as a guideline from previous experience with FDA approvals, whether it's a CDX or not.
That's right. And we generally set those guidelines for acceptance criteria when we're doing a validation plan, regardless of the level of validation. And we do have statistical analysis that's applied to our pathologist scoring. We do multiple replicates of scoring, and both within observer and between observer with our pathologists, and then use that information to create statistical values around the concordance, because they're ultimately the arbiter of that test system, right? The score itself is the product of that test system, not just the IHC staining itself. So, and yes, we use the 90% concordance that's recommended by CAP for both internal validations that we do, and then up to and including CLIA. These are great questions.
Yeah, they were definitely. So a big thanks so much, Christie and Kennon, for sharing your insights today, both in the presentation and the great discussion session. It's been a pleasure having you with us.
Thank you, Liam.
All right, a big thanks also to the audience here for participating. And in closing, we hope you enjoyed this Precision for Medicine and Scientist.com webinar, and we'll see you again next time. Have a great day, everyone.